Hi everyone, and welcome back to another episode of Syntalks, a podcast which explores the field of synthetic biology. This student-run podcast is organized by the iGEM Toronto team, an undergraduate research team hosted at the University of Toronto focusing on scientific research in the field of synthetic biology. I'm Kai, the host of today's episode, and I'm joined here by Amy and Henry, a member of iGEM Brazil. Can we begin with you both introducing yourselves and your role in iGEM Toronto and iGEM Brazil, respectively?
Okay, yes, like you said, hi, my name is Henry. Actually, it's Henrique, but no one actually can say it, so Henry, I think it's easier. I'd like to start to say I'm really happy to be here. Thank you for receiving me. So, I am a member of the Brazilian teams. We don't have properly a hierarchical structure. It's kind of anarchic in some way, but I've been a member of this team since 2017, so it's my third year, and I basically work in a little bit of everything. I help with, since I'm kind of old in the team, I help a little bit in the wet lab. I do mostly the dry lab part, and also in the human practice I help with some stuff.
Okay, thank you. And Amy?
I guess who I am. So as Kaya said, my name is Amy. I am the president for the second year in a row for iGEM Toronto, for iGEM Toronto's team. I've been on this team or as part of this team also since 2017. 2016, 2017? Yeah, so I've been part of 2016, sorry. So this is my third year as well. I guess 2016, 2017 overlap. Regardless, I've been here, same as Henry, I've been here for 3 years. My first year I was the wet lab manager, and then last year I was president, and this year I'm also president. So like Henry, I have my hand in everything with my particular role. I guess iGEM Toronto is slightly structured differently from what's up in Brazil. Which specific Brazilian team are you from? Sao Paulo?
Yeah,
okay.
University of Sao Paulo, the capital, because there are some other University of Sao Paulo teams. There are other two. One from Engineer School of Lorena, and another from São Carlos. So we have 3 teams in University of Sao Paulo, actually.
Yeah, iGEM Toronto is just iGEM Toronto, but we're from the University of Toronto, despite there being more than one university in Toronto. But no, yeah, so we structure our team a little differently. I guess to background it a bit, iGEM Toronto is made up about 35 people this year, which is about the average size of every team each year. And we have our administrative side, and then we have an R&D side. And on the R&D side, we obviously have our wet lab, dry lab, and human practices teams. And we all work together to create our project. So I guess that's good enough for an intro? Okay.
Yeah.
Okay, and can you both please give a brief description about the overall project that each of your teams is doing for iGEM 2019?
Okay, I guess since you started, I started. So this year, iGEM Toronto is basically, we're working on trying to optimize this little bacteria called apetase, and it was found 2 years ago naturally in this bacterium called Idenella sachiensis. And what this particular protein does, or I should say, this protein petase does, is it breaks down PET, which is a main component of plastics. And so we're trying to optimize this particular protein in terms of its structure and its catalytic activity and its thermostability in order to better break down plastics compared to its wild type. There was a paper last year that got put out by, I don't remember which lab, which is really bad on my part, but it was a paper that was released early this year. I apologize. It's early this year that had new mutations found in its catalytic site that helped increase its catalytic activity, and we're basically using that as a starting point and then further continuing these optimizations. And how we get these mutants are through different, this is where our dry lab component comes in, where we're exploring different machine learning algorithms to try and create these different mutants for both thermostability and catalytic activity. And then in terms of our P&P, or our human practices portions, we're really trying to focus on the idea of promoting better plastic separation and waste disposal and not only that, but also like the reuse of plastics. And we're doing it in many different mediums, not only through a panel discussion. We've also gone to a camp for younger kids, but we're also doing an art gala to try and get students, like almost all different parts of society, involved in this sort of conversation. And then I guess I'll just pass it on to present.
Okay.
Henry here.
So this year, our team is working with some LED light induced system. So we are using a system that a paper from Christopher Voigt's team published last year that basically describes a couple of systems that work together and that can detect LED light, all the three different colors, and differentiate among them. So what basically we are doing now, we are working with some of those systems. We are focusing in the blue system because it's kind of easier to work, and also we probably wouldn't have time to work with all the three. So we are using them to compare to chemical induced promoters that people usually use in bioproduction and things like that. So we want to describe and compare how this light induced promoter works in comparison with chemical induced promoters. And also we created a genetic system that basically detects light and then can turn the memory state. So you put the light, you induce a DNA change state, and then starts to express something. You put again the same light and then turn over to the other side. It's basically like a beat from engineering. So this seems to be working very well right now. And in the part of the human practices, we are trying to focus also on the bioproduction. We are trying to connect with some stakeholders in Brazil that work with bioproduction, things like that. They have some sort of bioreactors. Also, some of them already work with LED light. So we are trying to discover how we can improve the bioproduction by using light. Since you don't need to do any kind of purification after the chemical inducing. And also we are probably trying to make some other stuff in the public engagement part. We're trying to right now make a game, and probably will help to create the first high school team for the 2020 competition. We don't have any high school teams yet in Brazil, and we are trying to connect with some schools to try to create this first team. We are very excited about it.
Similarly, there's no high school teams that exist in all of Ontario either. So
Okay.
there's talk of some, but it takes a little bit of time to create. But regardless,
Yeah.
we're I think all of that motion of moving forward.
Okay, thank you. And now just to get back to the basics, if you think back to like 2016, 2017 when you joined, do you remember why you decided to join iGEM?
I started to know iGEM in 2016 because we are trying to create. I was in another degree. I was in another course, molecular sciences. Right now I'm in biology, and we were trying to create a team in this undergrad course because we found this competition about synthetic biology. I barely know in the time what the hell was synthetic biology, but it seemed to me like those stuff that you can manipulate genes and do things, and seemed very interesting. So in 2016 we tried to create. We didn't created this other team, but the University of Sao Paulo team already existed. So in 2017 a friend of mine was already part of the team, so I decided to enter in the University of Sao Paulo team, and mostly because it seemed very fun, seemed to be something very interesting. It's always been something that I was very interested into, this gene manipulating stuff from the high school point of view. I always been interested, so when I managed to enter it was really good. I think this mostly is because I looked at it and saw, okay, this might be fun. It's weird. Most of the things that I do is do because of this. I think I look at this stuff and say, okay, might be fun.
I guess for me, I joined back in 2016 because at the time I was entering my third year and honestly, I was looking for research experience and I saw this ad over Facebook saying we're hiring, and I was like, great, another research experience. I'm going to go for it because I don't know if I'm going to get it. And surprisingly, I got it. And I guess the reason why I went for it, just to give a little bit of background, during my undergrad degree, I had two majors. I had a major in neuroscience and a major in cell molecular biology, as well as a minor in physiology. Don't ask me why I did all that. Answer is I'm crazy. But having the cell molecular biology, I really, really like neuroscience, but I wanted to reach out to the different types of research fields that there are just to get my hands in almost everything so I can not only learn skills, but also learn what kind of research was being done. And so I thought that this was a great opportunity to kind of focus a little more in my cell molecular biology degree. Not really entirely cellular molecular bio, but still related, and then I can use the knowledge that I've learned here in an applicable way. So that's why I joined, or that's why I applied. And then when I got through the interviews and I got to join, I worked a crap ton in the lab. And it was a lot of fun, and I really, really enjoyed it, which is why I've stuck around for the last 2 years. But now taking on the leadership role to try and really improve and build upon what iGEM Toronto has already been in it, like what it already has been, and then making it better for the future. Because we've been around for a while. We've been around since 2005 in a combo team with the University of Waterloo. We were called Blue Waters, go figure. And then in 2007 we broke off, became a little more independent, and that's when we became iGEM Toronto. Now the team over the years has changed, but every year there's always room for improvement, as every team will know. And so I took it upon myself these past 2 years to try and better the way we hire people, the way we run the team, and the way the whole year kind of goes. So I've had a lot of fun so far, and I'm excited for the future as well.
Okay, this one's just kind of a fun one. Can you both please describe iGEM in 3 words, and you can expand on the words if you want.
Okay, I gotta get. Can I say good and bad things?
I think so.
Okay, I gotta think of this. Hold on. Only 3 words. All right.
We can try to do 1-1. You say 1, I say 1.
Hell is my first one.
I would say opportunity.
Oh, the difference.
Educational. I'll go good now.
Good. I would say headache.
Okay, third word. You said already said opportunity. If you keep going.
Okay. The last one is, I think it's future.
Future. I would say career. No, I'm gonna say a phrase instead of a single word, but student development. I would definitely say student development.
Do you think that is interesting? We just quickly discuss about these words. Why choose them? Okay.
You want me to go first? Okay. So for my 3 words was hell, educational, and student development. Hell because it's tough. Like I guess as every iGEM team will know, or anyone who doesn't know, iGEM runs on a very interesting time scale, and basically you only have 1 year to produce a project and then push it out. Whether or not you continue this yearly by year, like if you continue the same project over years, that's fine. But like with iGEM Toronto, we generally start a new project every year, which means we have to start from scratch, from project development to its execution within 1 year. If anyone has been in the field of research, they know this is hella difficult. Plus running the team, trying to get our budgets, trying to get the funding because iGEM is not cheap, trying to get enough people to stay because we don't have enough money to pay our students. So this is all volunteer based. It's a lot of work and a lot of organization, especially with my position as president and trying to make sure that the team actually stays afloat, not only within the school, but also following all the iGEM guidelines, meeting their deadlines and everything like that. It's not easy, and I really thank my team for kind of being here and doing their work and really investing their time into it, because without them I would have no team. educational because it is a multidisciplinary subject. Like synthetic biology to me includes almost all fields combined. You can put everything into the subcategorized under this, and it applies to different things. So many people learn new skills and they gain new opportunities, as you had mentioned, in order to learn not only about the subject itself, but also the interdisciplines within the subject field. And then obviously student growth, because a lot of students not only learn how to run their own research project and kind of feel out how it is to be a researcher, but also then, you know, what it's like to work on a team, what it's like to time organize, like it's both like the hard and soft skills that they've learned in an academic and non-academic setting. So a lot of this is like super, super important, and that's why I feel I mentioned like this student development part.
Yeah. For me, basically opportunity was kind of mixing together the education and the student growth part, because honestly I think the iGEM is a very good, like I said, like you said, it's a great opportunity for everyone. It's an opportunity where you can develop your own research, you can grow a person, like you need to work in a team, you have a lot of different persons, each one of them works differently, so you need to learn to work with all of them, and also you have opportunity to create something new. We have opportunity to participate in a competition, and honestly the jamboree is really amazing.
Oh, it's a lot of fun. I love it.
Yeah, it's a great opportunity not only for connecting with a lot of people from now around the world, but also you can connect with people, for an example, from Ginkgo by Works. That is a huge company. It's amazing. That was created basically from iGEM. So I always think that iGEM is a great opportunity for all of those things. The headache is basically for the same reason. In Brazil, we do kind of the same stuff. Usually we start the project in the beginning of the year. So every year it's kind of a new project. So I'm not sure if always this is the best idea that you have, but
it's so difficult sometimes, but I feel you.
But it's always, we have always some problems. I think that you said it's a very, very expensive thing. The money is always a problem. A thing that a friend of mine, she always says that basically it's a game for rich people, because it's really hard when you don't have money.
Yeah, I agree to that.
And also the organization doing this stuff, and molecular biology never works like it should be. In theory, it's beautiful. You say, okay, we do this protocols and this will be amazing, and then none of them work. So it's always problematic, and also future, because honestly I feel that synthetic biology is one of the most amazing things for the future. I think this is a really it is a thing that in the next 10 years can become of the main fields in the world, especially in terms of connection with the population and things that can really turn things for everyone. See, not that all the other stuff are not important. I'm from ecology, so I really love theoretical stuff, but I honestly think that synthetic biology, it's a great part of that fourth industrial revolution, this new revolution that the world can make. And honestly, I think this is a great thing for everyone. It's a great opportunity. Like again, it's a great opportunity for everyone, for the whole world to develop as humanity. Maybe I'm being too over here, but I honestly
no, it's a good way to develop the
yeah.
synthetic biology community for sure, 100%.
And what do you hope other members from iGEM Toronto and iGEM Brazil will take away from their iGEM experience?
That they come back and they learn and they be independent researchers at like different stages in their life, and like one of the big things for us at iGEM Toronto is we really try to strive for independence, because a lot of projects, especially when you go to the field of research, when you start off as a master's student, or even if you go and do a master's or like a research project as an intern, it's often a professor or a mentor gives you a portion of a project, and then that's what you do. You don't really have a say in your own development, i.e. it's probably because of time, but like there's a lack of freedom for you to choose what you want to do. And so like you lose this ability, like not lose, but you don't get that opportunity to learn, right? And in classes, you often don't do that either. It's mostly, here's a list of instructions, now follow. So great, everyone becomes followers, but in order to become independent researchers, you need to know how to develop, how to analyze, how to problem solve and troubleshoot. And so we try to provide that opportunity at iGEM Toronto just because our entire team is made of undergrads. Solely the R&D team is entirely undergraduate, from first year all the way up to fourth year or fifth year, depending on where you are in your school year. Administration, and we have graduate advisors that can be grad students. Like myself, I'm a first year's master's student heading into my second, but I have no part really in the actual development of the project. It's literally me saying yes or no, this is doable or not doable, depending on our funds and depending on our time. So what I really want students to get out of this is to have not only fun doing research, but also learning how to do research and how to work with other people in different fields to produce a very good scientific project.
Yeah, this is a fun stuff because when I look to the Brazil team, it's a little bit different because we have people from the last year in the masters, and we have people from the second year in their undergrad. So it's a really big range of people in different times of their careers. But I honestly think it's still the same feeling that basically we want people to learn. Not all of them work with synthetic biology. Not all of them work with doing an independent project. And honestly, I feel that one of the main stuff that I would like people to learn from this experience, it's also not only how to work in science and how to do science, but also how to do science with other people. Because a lot of stuff, people always think there's still this image of the scientist that is this weird person working alone in their lab with their crazy minds, but this is not how science is done anymore. Science, the 19th century, it's a collaborative work. You cannot do this by yourself. You need all the other people for doing it. And I think this idea of you need to do a collaboration in iGEM with other teams, it's also a great way to try to improve this. So I think this is a thing that I would like people to take with them, this idea of working together and how to work together, and also of course how to manage a project, how to think about the project, how to think science, because this is not trivial at all. This is not something you learn in most of the classes that you take.
Totally agree.
Should be, maybe. It should be. But it's also hard to learn in classes. It's hard for the professors to do something like that. So yeah, I think it's kind of the same stuff that
Okay, and if you could change or improve anything about iGEM from like your own team's project to how it's run locally or globally, what would that be?
Oh boy.
Oh jeez. I'm gonna skip and I'm gonna avoid the topic of finances because that's too complicated and it's not fun to talk about because I'm probably going to get angry and I don't want to get angry. So other than shitty finances that need to be improved upon on both not only within our team but about the organization and then just how we get funding in general from the universities. Another thing that maybe I would change, or actually I guess I could say that isn't changing, is right now how we're doing something simple, is a local thing to our team only, is simply team recruitment. Usually we turn over our, for example, our 2018-2019 team from this year, or the one that I'm currently doing, the turnover would generally happen. We'd get our new administrative team at the end, so after the Jamboree is over, so in late November, is when we get the new 2019-2020 executive team. And then in 2020 do we then get our R&D executive team and our general members to form the rest of the entire iGEM Toronto team. But by then you're looking at somewhere around March, which is when we have the entire team together, and that's when we start project idea bouncing around, which is kind of late because then once you only have like exams and you run into the summer and then you have exams and right after exams it's just bam. But is there enough time to do everything? Maybe not. So what we're trying to do this year and what we're changing this year actually is we're going to be getting all of our executive folks, both on the administrative side and the R&D side, at the end of November. So that way they start their project development earlier in late November. December is always exam time, but like they can do it upon themselves, and then back in January once we get the vote right off the bat, and then we can get our general members afterwards. And hopefully with the shift in change, we get better candidates, more candidates, and we increase the possibility of starting sooner, which is always a nice thing. So that's one of the major changes that is going on right now. I'm busy trying to get everything put together in the last 2 weeks of September, or August before September starts. Woo!
Yeah. It's kind of the same too.
Yeah.
We are weirdly similar.
I guess we're similar situations.
Yeah. But I think that looking at the particular level, at the level from University of Sao Paulo team, I think that I think we need to change also kind of our organization. Like I said, we are kind of anarchic, but this also happens because we are a small team. Even that the teams already have started in 2012, so it already has 7 years of iGEM, 8 years because you count 2012. So it's the 8th year of iGEM in University of Sao Paulo, but we still have a lot of struggle in organization. We have some differences between our time in Brazil because we start our year in the beginning of the year, our university year.
Yeah.
So we also struggle a lot to get people to participate in iGEM. We only have 10 people working the team right now, so we are small, but we want to change this stuff for the next year maybe, and so we start to organize the iGEM year in January, maybe February, because our classes only start in February at the carnival. So maybe we'll try to start this before. We want to start this right after the Jamboree this year and try to get a better way to call people from iGEM. Right now in our team we only have people with a biology background. We don't have any engineers or mathematicians. I'm from biology, but I'm the people with the most mathematical background, and I have to say it's not so great. So honestly, I think that we need to improve, and I also think that now looking in a more vast point of view, I think that we need to change a little bit. It's the connection between the teams in Brazil. We have a lot of connection, but I honestly think that we need to improve. This year, we wanted to try to create a communication with the federal government, maybe trying to have federal fundings from all the teams, but right now there are difficult times for research in Brazil, so we didn't do that. But honestly, I think, especially from the teams from the same university, we need to create a better connection between us all, and maybe improve the help that we all do with each other. And maybe in the iGEM part, in the competition part, is more difficult to say, especially also because I don't want to get not angry is the proper word, but with the funding part and all those stuff, but it's really difficult and sometimes. So from the whole competition point of view, it's kind of hard to tell, because some of the stuff not only depends on iGEM, but sometimes depends on the government. Mm-hmm. So it's kind of a wall that's really hard to work with.
Yeah, I would actually like to ask you, because I guess our political situations are different in Brazil, but even within the university, mm-hmm. How is your, with Toronto, our Toronto team, speaking of funding, but just not so much on a global scale, more locally.
Yeah.
At iGEM Toronto, we are every year in order for us to get funding, we ask their departments. Now, some departments are very consistent and they support us. Oftentimes, because we have students from their departments, like at least one or more. I would say, not always two. It's one or more that we get from the departments, and they're willing to support us. They'll chunk out funding. But then there's other departments who one year will say yes and give us $500, another year say no, give us nothing, and then another year just randomly give us $1,000 and we have no idea what goes on. So the support within the university itself is very scarce, and we're constantly like asking. It just seems like we're just scavenging for funds. I don't know if it's the same at the university, because you have 3 teams within the university, obviously across different colleges or departments, however you divide it. So how is your local support in terms of funding? Like, does your university identify you as a legitimate research team, or is that different?
The part of the legitimate research team is a little bit complicated. So, but we have some support from the university. Even that we have 3 different teams in different campi, because one of our team, it's in the capital, is in São Paulo. The other are in 2 other cities, in 2 other campi. So from the university point of view, we have some fundings. This year was especially weird because we had a little bit more funding. So basically in our university, we can divide in, not exactly in departments, we have the institutes, but we have the supra institution, that is the University of São Paulo, and then it's divided in 4 parts. It's the undergrad, overgrad, research, and extension, that is basically connection with the exterior world, is connecting with the population and all this stuff. As you may imagine, this part of the organization, the connection with population, is the weakest one, the one that has less money. But we have fundings from all of these 4 organizations, departments from the university, and we also have some fundings from our institutes. So this year, the Institute of Bioscience gave us 9,000 reais, that is around 3,000 Canadian dollars, and the Institute of Biomedical Sciences gave us around 2,000 Canadian dollars, and all the other parts, the team registration fee and some of other fees, like the student fees, also come from this super institution that is the University of São Paulo. But this is a kind of interesting part, because year by year we are getting closer to these institutions. So we are getting closer to the University of São Paulo. We having better connections with the people that has power to decide the stuff. So we can talk to them about the funding and all those stuff. So I don't know how it works here. If you can talk with what's the name of the people that rules the university here?
So we have presidents, but like, our president obviously rules everything. But with we do have like a dean. I think they're called the dean of research or the president of research.
In Portuguese, is reitor. I really don't know how to say this.
If I knew Portuguese, I would be able to translate, but I'm Chinese, so obviously I know Portuguese, duh. No. So we've actually talked to our vice dean about it, but the problem is that their funding, they already redistributed to different systems within the school. So in order for us to reach into that funding, we have to flow or join the school system. The problem is the way our team runs does not fit into any part of those school systems, so then we're still left with nothing. So it's hard. Like, I guess we're in the same boat where they just have different systems that don't exactly suit iGEM folks, and then we're all stuck together with no money.
I think that sometimes happen like this supra institution that is the University of São Paulo. They can give money that we can use for all the people, but if we get money from Institute of Biosciences, we only can use for people that studies biosciences. If you get money from biomedical sciences, only people from biomedical sciences.
Makes sense.
But we also have and a funny part is actually our or PI, it's from the pharmaceutical sciences.
Oh.
So all the money goes to the pharmaceutical sciences. So every year we have some struggle to how to use this money and see the bureaucracy. Sometimes it's the worst part.
Yeah,
It's really complicated. We have tons of rules about how to use this money and how to really manage it. So.
Yeah.
But the problem it is, I really don't know the situation within the other University of São Paulo teams.
So you only know yours. Not
Yeah.
It'd be interesting to understand what happens there too.
Yeah, as long as I know, we kind of have the biggest fundings, the biggest grossings in the University of São Paulo. I'm not sure if the university gives money to all of them. This is a thing that, if I remember correctly, there was another team from another country that received some money from the University of São Paulo, but they didn't manage to participate in iGEM due to some bureaucracy problems. But right now, the São Carlos and the EEL teams, I think they may receive a little bit of money from the university, but mostly from their departments. It's a little bit different. How this works, and this is one of the reasons that at the beginning of the year we wanted to create a federal funding for iGEM. We were thinking about trying to reunite all the Brazilian iGEM teams and try to send a request for the federal government to say, hey, this is a big stuff in research. See, this is a big project. See how many projects, how many prizes we have won. So it will be amazing if you could give us some fundings. But again, it's a difficult time for research in Brazil. So, but luckily, in the last 3 years, we managed to pay and participate in iGEM. So.
Okay, and just a final question, just really quickly, just for the interest of time. What is a synthetic bio topic that you would like to further learn about, and why? And your knowledge of it can range from little to expertise, and anywhere from medicine to the food industry.
Okay.
Another topic of synthetic bio. Interestingly enough, I want to try and... I know this is one that's very far off, but since I study neuroscience, one thing I find would be very interesting is to combine synthetic biology and neuroscience and then coin the term neurosyn. But that doesn't exist yet. And I think it'd be really cool because, again, synthetic biology creates a lot of different techniques that can be studied and used in different fields of science. So, I mean, what we study, because with the genetic manipulations we do in cells and E. coli and yeast and even plants, you can apply the same sort of theory in human cell lines and eventually even towards neurons. But the problem with neural-based studies is that there's a lot of unknown things within the field of neuroscience itself. So trying to even apply these techniques to this this field of questions is difficult. So process, but maybe exploring that would be very, very cool.
I think for me, funnily enough, it's kind of the same thing, but in another area, because I work with evolutionary ecology.
Yeah.
So a thing that I would really like to see is using some of those synthetic biology techniques, all of those synthetic biology knowledge that we have right now, to try to develop some tests and also develop some ways to do some laboratory testings in ecology theory and evolutionary theory. There's a professor at University of Laval that worked with this, with synthetic evolution. The idea that you will try to do an directed evolution in a bacteria, but use the background of synthetic biology in this evolutionary theoretical part. What is a very interesting thing, because usually the knowledge do the opposite path. We start in a theoretical way. We start thinking about how things work, and only with some presumptions, and we do some arguments, and we create some hypotheses, and then we are going to the applied part and things like that. But an interesting opportunity and an interesting way to approach this, it's take this applied area that is synthetic biology and bring some stuff to the theoretical part that is evolution and ecology. So this could really bring new ideas, and also kind of medical part. But this is a thing that I don't work with, but this is something that I'm really interested with. The way that synthetic biology can improve the quality of life of people. Not only it's now thinking about ecology too, we can think about how synthetic biology could be used to improve like farming and things like that, and turn into a better ecological activity. We could improve our crops, improve even cows and all those stuff to work in a more ecological way. So honestly, I think the synthetic ecology would be a very interesting thing, not only from the theoretical part that is really promising, but also in the applied part, because we can really change things.
Okay, so thank you to our listeners, and thank you to Henri and Amy for coming in today. This has been another episode of Syntalks.